it's there. So our next theme is to develop and evaluate genomic prevention and therapeutic strategies. And the question that's come up is, how can health improvements from the use of genomics be demonstrated? I think there's several areas that are commonly focused, or that we've heard from several groups. And the first one is how to assess the clinical and personal utility of genomic prevention and therapeutic strategies. And this is really, how do we use genomics to actually look at outcomes or the impact of genetic interventions or treatments? And the second one is select when to screen and undergo management changes. And I think there was an interesting comment made about familial hyperlipidemia, and someone yelled out, if they're under the age of 45 and they have a heart attack, we should be checking them for FH. And I think that really raises a question, why 45? Why isn't it 50? So I think how do we, in genetics with therapy, how do we actually change those or come up with good management guidelines? And then the last one is, how do we integrate genomic, environmental, and behavioral variation? And I think we've kind of seen that theme throughout the day, is how do you combine all this information? Because again, the clinician is trying to get through the day. How do you compile more information and distill it down to something that they can make a decision upon? And so I will turn the microphone over to, I'm trying, let me get through these. And I'll turn this over to the moderator, Bob Wildin.
Thanks very much, and I will introduce the first speaker is Aniwaa Owusu-Obeng from ICANN School of Medicine.
You can do it. Yeah. Okay. Yeah, so I'm one of those that didn't have slides today, but I have my notes here. So I have 5 minutes, and I'm just going to go straight into it. So in 2013, I was at Mount Sinai. We had implemented pharmacogenomics for 3 drug gene pairs. One was clopidogrel, the other was simvastatin, and the third one was warfarin. But in November of 2013, 2 studies came out, the UPAC trial and the COAC trial. And knowing our patient demographics, about 70% of them were non-European ancestry. So we had to pull back our warfarin alerts. Fast forward to this year, in March of 2019, the FDA has just recently approved a new drug called siponimod. And siponimod is used to treat multiple sclerosis or relapsing forms of multiple sclerosis. The FDA recommends genetic testing for CYP2C9 and gives actually dosing recommendations as well. So if you happen to be a poor metabolizer, which is the star 3 variant carriers, the FDA recommends against using this drug. However, they recommend halving the dose for intermediate metabolizers, and normal metabolizers get their therapeutic dose. Why is this a problem? In 2013, what we realized with the COAC trial was that there were about 25% African ancestry individuals in this study, and the study basically gave us a negative outcome, I should say, because they tested for star 2 and star 3, which are predominantly found in European ancestry individuals. This theme has been sort of running through our earlier discussions today, but I just wanted to highlight it again, because as we talk about using or developing therapeutic strategies with genomic medicine, it's imperative that we make sure that we are including everyone. And in 2019, if there is a new drug that has been approved, and the new drug is giving dosing recommendations for a condition that is also predominant in African ancestry individuals, African Americans, then this is a problem, because it doesn't give dosing recommendations for the other star alleles, which are the star 5, star 6, star 8, and star 11. And FDA has no recommendations for these individuals. So what if you're an African American patient who happens to have relapsing form of multiple sclerosis? You get tested, and it comes back as maybe a star 1 because you were never tested for the other alleles. And so the patient, the physician might start you on the dosing recommendation that's labeled in the drug label, which might not necessarily be the right one for that individual. So this is a very pressing issue, and I just wanted to highlight it. But how can we get past this? We need that patient engagement. I think earlier we've been talking about that, and I just have some very out-of-the-box ideas about how we can do this. I'm thinking about how can we engage social media to actually do this. And I look at other examples all over, and not just in healthcare, but throughout. Look at the whole span of iPhones, or the use of new makeup conditions. Sorry, I'm into those things. And so, and social media is a huge marketing platform for these things, and it works. If we need to engage underrepresented minorities, they engage them on the avenues that they're already being tuned into. Social media is one place. How about even using pop culture? I know that Angelina Jolie at some point got BRCA testing, but imagine if we had a Beyonce PGS test panel out there, and Beyonce is talking about this all over. You wouldn't need to speak too much. And why is this important? I believe that patients who are empowered will empower their providers, who will in turn adopt this into practice. How does this happen? Look at how the whole direct-to-consumer prescription drug marketing works. Drug companies are spending billions of dollars every year on marketing because it works. Yes, there are some cons to that, but if we do this responsibly, that our patients could be empowered well enough to then, whenever they come to the doctor's office, they're asking this question. Doctors will then have to be forced to learn about this, and also in turn adopt this into their practice. I think my time is running far, but I had that point one. And also I just wanted to quickly touch on this idea of also highlighting the real patient stories on social media, or whatever platform that we choose, maybe even Facebook groups. These work for other disease conditions, and getting patients to be informed, getting patients in some sort of advocacy groups. So that's my time, but hopefully we'll get to talk more later. Thanks.
Great. Do you want to introduce yourself?
Oh, whatever you want.
Go ahead.
First, I'll see how I can screw up advancing the slides. Ah, hello. Hi, everybody. Okay, away we go. So I'm going to be focusing on Carol Horowitz. I'm a general internist, health services, and community-based researcher, and I'm going to be focusing on chronic diseases. So the first question I wanted to ask you guys, we know we're hot, right? Genetics is hot. We're the bar of the restaurant. We're the belle of the ball. But the question is, this tide, are we going to float or sink the other boats? So if you look at the table over here, you can see you've probably seen this before, the CDC pie that shows where genes and biology are an important part, and this is where we are. There's a lot of other factors that determine people's lifespan and quality of life. So who might we overlook in our push to precision, and what might we overlook? So one of the things I wanted to point to in terms of what is, when I first started joining my genomics colleagues, when people talked about G by E, it was like genetics with hypertension. But let's talk about the big E's, like, poverty, racism, stress, pollution. Right now, I do a lot of work with APOL1, which is a gene variant that increases the risk of kidney failure in people with African ancestry. Well, air pollution increases the risk of kidney failure also. Lead poisoning does also. So I think we need to look at G by E studies, because that's going to make our work much more powerful, not just G by G, but G by E. I also wanted to point out this chart, because I think as we're looking at our perspectives, and I do think our perspectives need to change to enhance our partnerships, we should think about that we're here. We're in the members of kind of researchers and to a degree industry. But that's not what this is about. We're serving clinicians, we're serving patients, advocates, and communities. That's why we're doing what we do. So if we think about what we do, we think what we do is really important. The benefits are obvious. The risks, and that we think patients should, for the most part, listen to us because we know what we're doing. Whereas clinicians are going to look at the purpose and say, well, research is good if it changes what we do. And the benefit of genetics, it probably helps a small % of our patients, but it's one tool in a big toolbox. And the risk is, what's the time sink? What's it going to take me away from doing? And their focus is on patients. For patients, we have what's called earned skepticism. What is this really going to do for us? Is this going to be drive-by research where you come in, do your work, you leave, and we're still sick? And what are you taking the resources away from? Put them into housing or schools if that's all you're going to do. And the risk is unacceptable if the benefits are iffy. And they're feeling like their roles to question and contribute. And thinking about payers, that's a whole nother way that people think about this. So I think in order for us to move forward, we really need to think from multiple perspectives and remember who we serve. And I have to remember where I'm pointing to. Oh, see, I screwed up again. Okay, so the first, I wanted to talk, so that was my first point. The next point that I wanted to point out is how can we operate differently? How do we build equity into our work? Why is research so white? So you've heard this saying that systems are perfectly designed to get the results they get. So our patient population research is white because that's what we've been resourcing. So let's think about what we can do differently than have the slide I'm sure you've all seen about our genetic databases. What if we change what we pay for, and what if we instruct reviewers differently, right? No excuses. No excuses at all. It's not that it's too hard, it's too expensive, we don't have time. What if we had a minimum fundable diversity quotient with limited clear exceptions? There are founder populations that wouldn't work. But if African ancestry and Hispanic ancestry are a tiny part of our GWAS data sets, and they're this much of the population, is there something that if you don't get up to a certain number, we're not going to fund you? What about literacy levels? If you don't have 8th grade reading levels, we're not going to fund you. What about stipends? If you spend enough time with people or this much invasiveness, we get paid, you get paid. What about non-English speaking? Know what % of your population doesn't speak English. You can't have an English-only study if you have a largely bilingual folks. And finally, genomics exceptionalism. So we've done in our study with APOL1, we actually had genetic counselors train clinician extenders, lay people, to return the results. And people found them great. We offered everyone free genetic counselor. No one took us up on it. So I think we need to think about what we're doing and building ourselves as part of a larger community. Being careful in this Wild West, we don't overuse, that we're not causing false positives that are causing harm. And not asking patients to pay for things when payers aren't ready to pay. Let's get payers to change. We need to monitor for inadvertent widening of disparities, and I think we need to require engagement from the outset. Working with payers for coverage decisions, working with health systems, and disclosing things in patient-centered ways. And finally, let's give results back to patients. They deserve them. Thank you.
Whoops, that's not mine.
Next one.
Oh, you had another one. Okay, so I'm Patty Spears, I'm a patient, so I don't do research, so I took a little different stance. So I'm going to tell you a story. It's a story about Dorothy. Dorothy is a journalist, so she wrote about a story, and she decided to get her DNA analyzed. She had seen pop-ups on social media and seen it in her feeds, like we all do, and she decided she wanted to join the party. Right, and so she spit in a tube, sent it off to Silicon Valley, just along with 26 million other people do. And she thought that her biggest risk was privacy. And so, of course, when she sent it off, she checked all the boxes, keep everything private, and she was good to go. But privacy was not her biggest problem. So, you know, a little while later, she gets the email and says, your results are in, click here. And she clicked there, and it changed her life forever. So when she went there, the internet told her that she had a BRCA1 mutation. It was at a high risk of breast and ovarian cancer. Very unexpected for her. She cried. So what happens at that point? You have nobody to talk to. Where do you go? Dr. Google, right? So everybody goes to Google. You have nowhere to go for it. So this is a problem. You know, we're treating our health care and these major illnesses and our risk for major illnesses like it's a game out there. And this is happening right now every day. And so that's not the only problem. So one of the other things is that this test that tests for BRCA mutations from 23andMe only tests for 3 mutations. And we know that there's 1,000 mutations in BRCA1 and 2 that are actually put you at risk for breast and ovarian cancer. So a negative test does not mean that you're in the clear as well. So just because they tell you you're not at risk or you don't have BRCA1 or 2 mutations, that's not true either. And then there was another article, these are all within the past year, by the way. There's another article that you can be told you have a mutation, then go to the clinic, and Meg just told me of another mutation that somebody had that they went to the clinic and they were actually negative for that mutation. It was incorrect. And so this is happening all over the place, and actually it's getting worse. So, is there a placebo effect? So if you get a genetic test done and it comes back that you have a poor endurance gene, do you do worse on a treadmill? They actually tested that, and you do. So it has this placebo effect and changes behaviors and may not change it in the way that you think it will. It may change it negatively. And if you get bad news, and you're convinced you have a bad gene, it can really affect you socially. And so we need to keep that in mind. Also, I don't know if you remember, but the Golden State Killer was actually caught because a relative had actually submitted their DNA to Ancestry.com or one of those commercial companies, and they traced him through that. So when you give your permission to use the data and to use it publicly or whatever you do and to submit your DNA, you're actually submitting the DNA of all of your relatives as well. So you need to keep that in mind because this data is not kept in a secure location necessarily. And now, these companies that do these commercial tests giving out these medical information are actually now they want your health information, and they're collaborating with other companies like Airbnb for ancestry destinations. They've also connected with pharmaceutical companies and sold your data there. And they're promoting it. Okay, they're using the Grinch. I like the Beyonce thing better. And so I think that as we go forward with genetic testing, this is what the public is seeing. This is really making the public question a lot more. And so I think that we really need to get out there, and the professionals need to get out there with a clear message, a good message on how genomics can improve your health. And because all of these stories have been in the news lately, and that's what the public is going at. So I think a big education component for the public, for patients, is really needed. I think that, I don't know if NHGRI can do anything, but really this unregulated space of genetic sequencing that's giving out medical information needs to be regulated because it's not fair to patients to give them this information without a genetic counselor around. Thank you.
Okay. Hello. Okay. Hi, my name is Sula Hood, and I'm an assistant professor at Indiana University. And I am in public health, and my work largely focuses on creating community-engaged, not just community-based, but community-engaged approaches to enhancing genetic literacy. And I work in a variety of different contexts, and one of them is to work with families and in a generational capacity. And so today I'll talk a little bit about the power of genomics for prevention and why intergenerational family interventions are a must. Okay, so I'll start off with a story as well, and this is taken from a newspaper article that came out about the fact that a family reunion prompted genetic testing upon having communication between family members. And so a female relative, her name was Thelma, a female relative died from pancreatic cancer a few weeks before the family reunion, and she had previously survived melanoma. And her children told their extended family that genetic counseling might be a good idea. However, after an initial attempt, there was little interest. However, after discussing it further, her relatives realized that all of her siblings had also died from some form of cancer. And so once another relative was subsequently diagnosed with melanoma, over a dozen of their family members ended up deciding to get genetic testing. And a genetic, it was observed that a genetic mutation predisposed them to specific cancers. And so among the extended family, the physician identified 17 who had gotten melanoma, and 5 had pancreatic cancer, and 2 had both cancers. And so this just really emphasizes the power of engaging families, and when families are equipped with information and can help to actually not just look out for their own selves, but an entire family can be saved. And also it's important to note, when we think about the power of family health history information, it empowers patients to be able to communicate with their clinicians, which in turn informs their treatment, decision making, their screenings, but also it's important for younger generations as well. And that's one of the things in terms of prevention with families. I found within my own work, people are very, very protective of the younger generations and families. And so they are knowledgeable about things that might run in the family, and that can influence the behaviors of adults, but also kind of shape some of the norms and looking out for younger generations. And then this is just a little bit more from my own actual work, something that has come up several times today. We've talked about the importance of increasing diversity in this field of work, and I really am a strong proponent of engaging diverse populations in a context that is meaningful to them. And one of the ways that I work with populations is actually in the context of family reunions, which are a culturally and historically significant event within the African American culture. And so it's a large family gathering. This is, hold on. This is just an example of one of the reunions that my team and I have worked with to provide preventive information and then educate people about the importance of health history and increasing genetic literacy. But you can have up to 400 people, and they take place annually or biannually, so it's a potential opportunity to engage families longitudinally as well. And so I think it's important to think of these out-of-the-box and innovative approaches. And we've been very positively received, my team and I. Okay. So once again, I think it's important that we engage the community in a meaningful context, and we must provide this information in a way that is very clear to patients. And I think this goes along the lines of increasing genetic literacy. Once again, we have to be able to have patients that understand what we're talking about, and I think there's a place for public health in being able to do that. I'm almost finished. Okay. And so why should NIHGRI do that? That's one of the questions that has been posed. And so I think as a institute that is focused on cutting edge approaches, innovative strategies such as work and family reunions or other types of approaches are appropriate for the institute. And also along those lines, there are significant opportunities for scaling up, and that's something that we need to start doing more. We do these siloed approaches, but something like working with families, that's something that can be scaled up in a larger capacity. And then why hasn't it been done? Talking about health and families is taboo. And so once again, once families start to be more knowledgeable about the benefits of genetic information, I think that they'll be more open to the preventive benefits. And then this work is very complicated. Families are complex, and so many studies only have focused on smaller nuclear families or dyads, and families are much more complex than just 2 or 3 people, and we can learn a lot from intergenerational families. And then lastly, funding is needed to do this work, and I think this is an opportunity for us to maybe think about how we might shape funding opportunity announcements in order to be able to strategically have studies that focus on not just smaller families, but more large families where we can gather more information. Thank you. Okay.
Great, thank you. Is it on? Yeah. Okay. Thanks, everybody. When the talks here fit loosely under our charge, so I'm going to try to sort of bring that together and think about this as being examples of communication and dissemination of truthful information.
We can barely hear you, Bob.
Communication and dissemination of truthful information about genetic risk. And so that's kind of the frame that I want to put this in. I have some questions, but I want to give it up to the audience first.
Any immediate responses, thoughts from the audience?
So in terms of the potential horrors of 23andMe and all the things they could do by, such as GWAS for what we will be searching for on the internet and buying. It is a success story in terms of it's 20-something million people that paid to get their genetic data and volunteered to let their genetic data and their, I think they even enter their own phenotypes manually into the system. So I would say, like, what can we learn from 23andMe in terms of making a large-scale, genetic study that is easily funded because the people are paying for it, right?
Right. So I think it started out as a, kind of one thing, you pay and you get your genomics done and all. But I have found that over the years, and it might be good to learn from them, but they aren't sharing their data with the health teams. They aren't sharing their data for the betterment of all mankind. They're selling their data to get more money because they found it's a commodity. And the patient isn't benefiting from that. They don't even know that it's going on. So they actually just linked up with Roche Genentech, right? So now the data is being used to develop new drugs. They should be paying the patient for the data rather than the patient paying for the information that they get that one time. And now they're linking up with all different things. And so I think it's just a matter of really educating the patient on what can happen with their data and what can go on. And because they're not really being consented quite properly, I think it's for some other companies, it's like opt-out instead of opt-in, things like that. So I think there's a lot of communication that can go on. It's an opportunity to educate people, but I think right now, for them, it's a money maker, and so they're marketing it as that. So, but they do have a lot of information, but I don't know what analysis they're using.
Yeah, I'll just add to that the one thing I think we can definitely learn from them is the fact that engaging people with marketing, certain marketing things, I believe we should do it responsibly. I believe that we have a group of experts that can come together, put together information that has been vetted amongst our peers, information that we all agree with, and put that out there. And what they're showing us is if you put that information out there, patients are smart enough to pick you up on that offer. And maybe what we should be doing is putting that information and giving patients a right method or a right way to get these things done. Get it done through your healthcare system, through your doctor, in the proper channels that we in this room agree with. And I think that's one thing we can learn from them. However, with their strategy, there is a cost to it. So that can also sort of enhance these health disparities, socioeconomic disparities. If we chose to go that route, I'm sure there will be ways to fund that with research questions that are attached to it. So I think that's what we can learn.
So can you suggest any ideas of sort of scientific questions that can be asked in the category of dissemination science? So dissemination science is often linked with or grouped together with implementation science, which we talk about a lot here, but we don't talk a lot about dissemination science and how we educate, how we get the word out about studies, how we get accurate information out to people in the community about their risk. And it sounds like there are potentially a lot of different ways to go about doing that that may depend on the particular population that you're targeting. Particularly diverse populations may need diverse methods, I guess. So can you talk any more about that, any of you?
So I am a white middle-aged professional who works in Harlem and then drives home to the suburbs to barbecue. So for me, it's a little bit arrogant to think that I will understand a diverse community without having appropriate community partners. And what I've learned over the years, if I start building research ideas with whoever the target population is, clinicians, patients, advocates, at the table with me every step of the way, it's no longer my research, it's our research. So first of all the approaches we have make more sense because, like I said, who am I to engage a community that I'm not part of? And what we found is if we generate enough excitement and buzz in the community, and it's community partners who are going out talking about this, clinicians who are going out and talking about this, we are able to have that excitement. And then when the results come, and they're our results, they're at the table analyzing results with us, so they're the ones saying this is how the results should come out. So, you know, when I go around and ask researchers what they do if their research is wildly successful, what do researchers say? I write a paper and I write another grant. But there's not a lot of that obligation to go out to the community where you did the work and say, this is what we learned. If you do that, you start creating communities of people who are interested in research and don't think you're just using them for samples they don't know what you're going to do with. So for me, the partnership has made a really, really huge difference.
I also think it's very important for us to once again, I talk about meeting populations where they are and just the public in general. At the end of a lot of my presentations, I provide information, for example, about family health history toolkits that are useful for family reunions. A lot of them have been culturally tailored, and some that, for example, a group out of Utah put together that can be used with a variety of different families, and people are hungry for this information. They are excited to use this with their own families. They didn't know it existed. I knew it existed because I was, like a typical researcher, I'm writing a research paper about family reunions and health, and I know that this resource exists. And so we are going to have to be very intentional about the spaces where we disseminate information so that people know that it exists, so that they can then use it, and thinking about these out-of-the-box approaches to spaces, libraries, barber shops. Churches are widely used as well for data collection, but what about data dissemination as well?
I want to mention one other quick thing. A lot of times when we return results to clinicians and communities, we do it as a legislative breakfast, and we bring all the local legislators in also, and that's where the real magic starts to happen.
That's a great idea. I'll make an observation that Carla Easter and Vince Bonham in the division of, no, I can't remember the name of the division I used to work with. Education, right. So have been doing the sort of partnership kinds of things for quite some time. And so maybe, you know, the challenge is how do we help researchers out in the field add that to their toolbox in terms of engaging additional populations for the kinds of research that we're supposed to be talking about in this session. So other ideas from the audience?
I really love the idea of, you know, finding the right context for the right population. And I was thinking of that barbershop hypertension study that came out of Cedars last year, and you just mentioned barbershops, and it was such a cool clustered randomized trial that showed if you bring the pharmacists to the barbershops and have the barbers help with the health education and behavior change, you see a much more successful drop in blood pressure. So I would love to see NHGRI study something like that. We've had a lot of discussions today, or fun studies of something like that. We've had a lot of discussions today about engagement, both and behavior change, both provider and patient, whether that is different points in the lifespan, whether that is the nature of the clinical interaction. I think the idea of thinking about some totally novel, at least genomics contexts, is another really great way to think about engagement. And I'm just curious what other contexts other people think might be worth studying.
I'll say rural.
I mean, apart from the barbershops, I know we mentioned churches is a big one as well. I just getting people who are influential in the society to also, you know, be your champions. Like how we have physician champions when we are rolling out implementations. You have community champions or influential leaders champions in that regard. It's very powerful. And I would say it again, having celebrities do this, it will be powerful as well. And it works for different demographics, obviously. But if you do, I was telling Bob earlier today, if you target the younger folks in the family, they tend to be the caregivers. They tend to be the ones that are speaking to their parents about, you know, mommy, this test is good. Maybe we should look into it. Ask your doctor about it. So they are the people that we should really be targeting. If we are doing this in schools, I know Donna has, we mentioned maybe sometime last year, there are some high school engagements with genomic work. That's all important. These are things, start from that place and let the family disseminate it to their family members.
Yeah, I think it's really important to meet people where they are, both physically and also in understanding. And I think in using community partners, you break down those barriers. I think it was mentioned in one of the talks about health literacy and numeracy. All of that is very contextual. And I think that, when I did a lot of community outreach in North Carolina, and I always found a community partner, and the voice in the community everybody would listen to, Miss Ruth Royster in Roxborough, without her, you can't get anything done. So, I think it's really important to do that, and, both in the spot as well as, what you're saying to them has to be in context and appropriate.
So in doing this kind of engagement, which I think is amazing, the normal metrics of measuring success are not necessarily relevant. And so I'm just wondering what your thoughts are on what the appropriate metrics of impact should be.
So what are the measures that a researcher would be reporting out?
So I'm not sure I understand the question, because for me, this is an approach, not a method. So I do the same multi-site randomized controlled trials as other people, but my approach toward them is a partnered approach. So what it means is when I'm doing my outreach, I'm not just going to an EHR, doing a data run and sending letters out to people. So can you repeat what the...
So I guess I'm thinking not more in terms of promoting genomic literacy by engaging in these different ways and in these different places. How can you tell that it's worked? Not necessarily through a trial, but through... Is my mic not working? Sorry, I'm not close enough. Sorry. It's more just the literacy in general, as opposed to as a tool, as part of something else, but just genomic literacy in general. How do you measure success there?
So I'll just say briefly and then hand it over to Aniwaa. For me, it's really what's your primary outcome. So if you're measuring literacy, what are you looking at? Are you looking at a literacy scale? Are you looking at what somebody does? And then you just use engagement to see if that enhances the impact of what you're trying to do. So again, to me, it's an approach, but you still need your same primary outcome, whether it's literacy or getting screening or changing behavior.
Yeah, I was actually going to say the same thing. It's... What is the adoption metric that you are? Is it that at the end of the day you got 100 people in the community to get tested, or that there was a cascade testing rates that you're looking at. So you have to have those sort of metrics ahead of time, and then not necessarily objective assessments of their literacy. I don't think that's what we want when we do these community engagements. I think you should be looking at other adoption metrics. Yeah.
Mark?
Yeah, this has been a great discussion because I love the focus on the patient engagement, but I want to take it one step further, because what we're really talking about here is a continuum, and engagement and consultation is the second level of the continuum. The third level of the continuum is a true partnership, and I think it was alluded to, well, one of the things that got me thinking about this was the comment that I'm a patient, I'm not a researcher, and I don't think we should have anybody ever have to say that. You should be a patient and I'm a researcher. That's what we want to accomplish, because then what we're talking about is partnering. And so as we looked at the laundry list of maybe things NHGRI shouldn't fund if they don't have this, PCORI doesn't fund you if you don't have substantial evidence of patients as part of your project, up to and including as funded investigators. Now, that includes, of course, a commitment to training people so that they can participate in research, not turning them into researchers because we don't want to Stockholm syndrome them. We want that perspective that they bring to the table that's unique, that makes the research actually applicable. And we've had some success with this, and you know, as researchers partner, and I think we've been hearing the stories from you as well, you learn things that you would never have thought to even ask if you don't have that type of partnering. So there was an example a couple of years ago where PCORI, and I think it was the National Institute of Aging, actually issued a joint RFA using PCORI methodologies, insisting on PCORI engagement. I think something like that, where we really are looking at patient partners, patient investigators, to answer some of the questions that we've been talking about, would be really interesting.
So I'll respond to that. Since I said I was a patient and not a researcher, I'm actually a researcher, but I'm on this as a patient perspective. So I'm not going to give you methodology. I'm not going to give you bullet points. I'm not going to give you, oh, this needs to go there. That wasn't my intention being here. And that's why I prefaced it that way. You're not going to see what you saw before, because I'm not here to do the research per se. I'm on PCORI grants as a patient investigator. I'm actually a microbiologist by training in the research lab, but that's not why I'm here promoting genomic health and genomics to improve health. And so I'm not going to, like I'm on research grants a lot too, but I don't tell you what mouse to use, but I want you to go after an outcome that's important to me, and that's how I can influence the research. So it's a different type of thing. We don't bring the qualitative research that you're going to do the p-value and all that to the table, but I'll understand it enough so that I can contribute. But it's different, but that's a whole nother patient engagement thing. But I think it's important to have all kinds of patients at the table, whether you have me that has the background, or whether you have my mom who's just a patient, or whether you have, you know, somebody from rural America. You need to really have a lot of different influences in your research. So
one of the reasons I was poking at the idea of having, is there a minimum something that NHGRI should say, if you dip beneath this, we're not that interested, is that I think it's wonderful that NHGRI has enhanced diversity sites, but what that means is we have a lot of unenhanced diversity sites. And at some point, I think the enticement of funding could get people to change the way they do things and say, you know, unless you are in an area that has no diversity at all, and if so, you should find a partner with diversity, figure out a way to bring it in. There's lots of people around the country that can help you do it. So I agree that sometimes we want to do true community-based participatory research with PCORI, but there could also be times that NHGRI could say, and I think, you know, Caesar is a good example of that, we are requiring some level of engagement, some level of diversity, or this is not the grant for you. And I think the more we do that, the more I think people feel like engagement's it's too expensive, it's too difficult, it's too this, it's too that. It really isn't. Once you get used to it, it's not harder than other things that you do. So I think we have to push everybody toward it, and then people get more comfortable with it, and then the field will really move.
So I just wanted to share an example of a group that's really doing some pretty impactful work in terms of engaging communities of color, and that's the Personal Genetics Education Group, pged.org. As far as some examples of the types of communities they interact with, they do work with communities of faith. They also do outreach work at museums, libraries. They go to high schools in communities of color. And they do all of this to share, genetics that's relevant for the various stakeholders in those conversations. And it's really impactful work. I highly recommend you look at their website, pged.org, or go to their ASHAG talk that's coming up in Houston. And as sort of an example of how they organize their work, they are giving a talk around talking about race and genetics and how the two differ. And at the same time, during that trip, they're also going to high schools in the local area that have high Hispanic populations. So, this is just, I think, a really great example of ways that you can blend your scientific expertise with also engaging communities of color and other vulnerable populations.
Sir, there's one. Okay.
So I'd like to say I really like Carol's idea about sort of having a threshold or bar that you need to reach. In 2015, the FDA's Office of Minority Health provided guidelines for clinical trials along the same lines. And their idea was that the bar or threshold should reach the general prevalence across diverse populations. And so if there's a high prevalence of the condition in a certain population, that should be considered when you're designing your clinical trial and who you recruit. And so I think some of that can be borrowed from there. But other things that could be required. And I think that maybe I think the goal was also audacious challenges. And so I think one of the things that will also be required are human capacity building components to each of our grants and proposals. I think one of the things that I encounter when I engage communities, specifically around precision medicine, is people want the data or the research to come back to them, right? And it can come back to them in a couple of different ways. And actually, often they're not really looking for the individual report of different variants. They find that unique, and early adopters can like how it relates to their individual health, but they're really looking for impacts in their community. And so when we look at impacts in community, human capacity building, education, which is one of the questions that was put out there, education for underrepresented communities winds up being really valuable. High school pipeline programs that teach kids about molecular biology, that opens up the workforce, opens up pathways outside of certain barriers that exist in communities for having a diverse workforce. I think communities really value. And so when communities see, oh, my daughter or son or nephew is learning about molecular genetics in their summer program, and they're doing research, and they come in, and the whole family will come to do a poster presentation. And it seems like a small thing for lots of communities where a poster presentation in high school is sort of like old news, but in some communities that's a really big thing, and then you show your poster. I had a student this summer who said that every conversation that they had at their dinner table, that her father told me this, was all about genetics, and now that's all they talked about on their family chats, because families chat, right? And so I think that human capacity building and ways in which we can think about embedding that in our research grants should be a required component of what we do if we want to reach communities. The other thing that Carol mentioned, and I just want to make sure it doesn't get lost, as we prioritize diverse research samples, I want to make sure that we're not limiting that to just having diverse samples with the same questions that we were asking before. I think one of the key things that makes looking at doing research in Harlem and looking at APOL1 makes sense is because kidney disease was one of the things that was important to that community, right? And so we can't necessarily just have a diverse sample and have this really large national database or these big studies where we have diversity because other people have recruited them, and we ask the same questions, and we improve our tools, and we improve the things we're looking at, but not making sure that we're bringing the questions that are important to communities back to them. And so I think we need diversity in thought, which also goes back to diversity in our workforce.
All right, I have another question. Oh, yeah, go ahead.
And one of the things that I really stress and push, and I work in an academic institution, is really training the scientists, and the clinicians do it better, but the scientists really how to communicate their science to the public in a way that the public can understand, because otherwise they'll get their messaging from TV and other sources. So it really has to come from the scientific community.
And I thank you for your comments. I wanted to give you two things that we've done that are interesting. One on the research side and one on the dissemination side. On the research side, if you were to say, we need to know what % of your population doesn't speak English, because if it's high enough, you can't have a unilingual study, that means that researchers actually have to figure out what % of the populations they're serving don't speak English, which people don't know, so you can kind of hide under that rock. So that's an example of how making researchers respond to something changes what they do. On the dissemination side, one of the things we've done around schools, and I think this is available nationally, is that public school teachers need to get P credits in order to keep their education going, and they often cost money to get these extra credits of work that they do for training. What we do is we give them free P credits by coming in and teaching them about things. So we teach them about environmental health, and we're starting to teach them about genetics. They come in the summer, we teach them about genetics, and then we go out back to the schools with them. They teach the kids about it, and then we teach the kids how to do research. And that's a way to really start getting these ideas into the community, besides getting really great pipelines of diverse people into the sciences.
Questions? Yes.
I want to ask Sula about her experience with these large families. Do you have any empiric data on specific variants or diseases you studied? And then secondly, how do you account for the family dynamics in these large collections? Because I assume that plays an important role. And then finally, do you think there could be digital tools that could be used, like apps, for families to share data among themselves?
So in terms of the conditions that we're doing, first, we are just initially, this is kind of still very formative, and so we're more so providing education in terms of prevention on specific conditions. And one of the things that we've done is ask families when we're partnering with them if there are specific conditions that they'd like to have information about. And so we provide kind of information that I guess pertains to conditions that African Americans are more at risk for, the typical conditions, diabetes, and cardiovascular disease and things of that nature. But also one of the things that I have found in quite a few of the families that I've been working with and participants that I'm working with in terms of about their health history is lupus. That's something that is increasingly that I'm finding that families are interested in having more information shared about that. Your second question.
My question was about whether you think that sharing among family members could be enhanced by additional tools, like if you had a mobile app that might enhance communication of risk to somebody who couldn't attend the event and was geographically distant. One of the things we've noticed in family screening, particularly cascade screening, is that because of HIPAA, you cannot directly contact relatives. And so, in addition to giving a letter, have you thought of other means that they could, it'd be easier for them to share disease risk information?
Yes, and that is actually the hope of a lot of this work, is that we are equipping families with the information that they can then share, not even just with their own family members about their own health history, but the information that we provide in their larger networks as well. And so adapting some existing materials for use in this context and making those available. One of the things that I enjoy and have found in working with family reunions is that it's a setting where there's a lot of social capital, and so we can leverage the existing resources. So oftentimes we might find that a family has a nurse or a physician in the family, and they typically are the people in the family who are trusted individuals that everyone calls when they have an issue, but those individuals can also be engaged as kind of like health champions within the families and start to get the families involved. And once again, I think for something that's a tradition that is regularly occurs, such as family reunions, this is something that can be integrated into the tradition and into the practice that occurs year after year or biannually with the families. And so we do provide resources, such as, the slides. There's also toolkits where they can where you can train people in the families to facilitate these discussions on their own. That way, in the event that someone isn't present, they still benefit from that information that we provided. And then I think your third question had to do with unique approaches to I guess being able to engage these families and with that information. One of the tools that families we have been promoting the use of and that families have found interesting is, for example, the My Family Health Portrait tool. But I think other apps definitely, as we develop more apps, will be very useful to families. One of the things I feel like someone else might have brought this up earlier is that families tend to be in strong communication, for example, outside of a family reunion. You saw that large family up there, but even outside of that, they have Facebook groups and all different types of things. And so we need to think about joining the ways in which families communicate. They have newsletters, all types of things. It might be a newsletter that has a family health component, for example, or information with the families. And so there's a variety of ways to engage them and leverage that communication.
We had been talking earlier about Facebook communication and other social media things, and whether social media is a potential target for work dissemination, basically, of both information, generally education, and family-oriented information, or whether can we get NHGRI to build a special social media app for this? So in my work, we have been using, the hospital has a group of patient family advisors who are essentially unpaid volunteers, and they have actually been extremely helpful in helping us formulate ways of implementing our testing projects. And I'm wondering if you've had any experience with that kind of embedding process, and whether that might play a role in some research.
So we've moved. We used to have patients and family advisors. The first thing we did is we moved from an advisory board to an action board because we didn't think people should be considering this passive. The second thing we did is that we changed it from patients and families and advocates to more of an accelerator, where around the team we also have device makers, app developers, industry, health system leaders, public health people, so that everybody is influencing each other and is influenced by each other. So that's been really exciting. And I think having the folks, no matter what you call them, is really great. The one place we would distinguish is that we don't have volunteers. We feel like if we get paid, they get paid. So we actually usually ask the group what they think is a fair amount as a yearly stipend or an hourly stipend, and we build that into the grants. And we've never had a problem with that funding coming through.
I know that in my own work, we've used community advisory boards as well. So I have a family reunion advisory board, for example, and they've been very helpful with thinking about sensitive ways in which to engage families and the materials and everything else. And so it's been very meaningful, and we couldn't do this work without them. Even myself, for example, when I'm engaging African American families and communities, I'm still an outsider. I'm affiliated with a university, and so in that regard, you know, we think about things such as distrust, and so it's been very helpful working with the community to be able to kind of overcome those barriers as well.
I've got to say, I really like your idea. We've got the Patient and Family Advisory Council at my institution, as well as I've got a patient research advocacy group. But what I've done is brought in the health literacy expert, the regulatory people, and all the different people, because I think unless you learn from each other, you really are just talking to yourselves as patients. So I think that that's a really good model to follow.
And I'll follow that up with a kind of a follow-up question. So another experience we had in going to the large hospital council, patient family council, to get their input on whether we should develop pharmacogenomic testing. And the response was, why aren't we doing this now? And maybe that's, you know, my gift of presentation, but but that was certainly something that made us feel like we were probably moving in the right direction. And the question is, with that sort of reflection from them, which was in some certain cases very strong, how do you, I mean, our role there was to gather their input, but how would you envision sort of translating that into action on their part to help remove barriers to move that along?
So I'll tell you my experience. We had a focus group of actually our eMERGE participants. We called some of them back, had a very high rate of response, and we actually had to turn some people away. But when they did come, these were eMERGE participants. Granted, they had been genotyped years prior, but they were surprised that they were still that small group and that it wasn't the entire health system that we were doing. So we got similar response. How come you're not testing everyone if it's so good and we're reading all these things on the media? How come it's not being done widely? And so what we talked about was that they should come back and help us develop patient education materials. So that was one thing that we put out there. We know you're very excited, you're enthusiastic about, you know, some of these approaches that and innovations that we're doing here. How about you help us? And so we, at that very meeting, as part of the focus group, we presented a drafted brochure to them, and they helped us tear it apart. I felt bad for the students that created it, but they tore it apart but helped to build it. Some actually gave us their contact information to come back when it's ready to give us feedback on the finished product. So I think that's a way to sort of turn them around and help to engage them for the betterment of the entire program. Yeah.
I'm often surprised that people are surprised that of all of the stakeholder engaged research I've ever done, the most interested stakeholders have ever been is in genomics research. But that's how I found it. And I think often people have an implicit bias that certain populations might be fatalistic or unmotivated or low literate or these other things that make people, and that's not true. So I think one of the things that we find with this interest is overcoming people's implicit bias of who is interested in what. And this is why I think when people reach out, you can get really good participation. And I don't think that the whiteness of our data sets is because people are fatalistic and mistrustful and not interested. I just think we're serving up something on the wrong platter. You do a good invitation to a good party, and I think you'll be successful.
Other questions?
Thank you.
Thank you very much.